I'm Romney Humphries. I'm a director of microbiology and infectious diseases labs at Vanderbilt University Medical Center. And I'm here today with Emily Bentley, who is one of our wonderful technologists in the micro lab. And Emily has some questions about non-fermenter susceptibility testing. So Emily.
Why are there no diffusion breakpoints for non-aeruginosa Pseudomonas species or non-enterobacterales?
Yeah, that's frustrating, isn't it? Because sometimes they don't grow on the system.
Yes.
Yeah. So the challenge is we evaluate all of these bugs under that non-enterobacterales breakpoint table in M100. And so part of the answer is how we set disc breakpoints. And so how we do it is we have our MICs, and then we test hundreds of isolates by disc and MIC at the same time, and we see where we can kind of get those to agree. And so this process is detailed in a document if you want to read about it called M23. So this hasn't been done yet for those non-aeruginosa Pseudomonas or some of the other non-enterobacterales, but we are working on M45 and we are looking at disc to MIC correlates for the non-aeruginosa Pseudomonas species, so Pseudomonas fluorescens, putida, and stitzeri. And we're also looking for disc correlates for Achromobacter. So let me show you what this looks like. So this is just one example of ciprofloxacin, and I think these are some of the non-aeruginosa Pseudomonas data. So we've got the disc zones on the x-axis, and then the MICs are on the y, and then the numbers in here are how many isolates had an MIC of 0.5 and a disc zone of 27. So we plot this all out, and we say, okay, well, we know our MIC interpretations are here, and then if we look at that, where do we draw the lines for the disc so that we minimize major errors? So isolates that have a really low MIC and a teeny tiny disc zone size, or very major errors, ones that have a big giant disc zone size and a really high MIC. And so what we really want to see is most of the bugs falling in here. So that's how we do it. And this just hasn't been done yet for all these bugs. And you really want to look at them like organism by organism, because what might work for Achromobacter not necessarily going to work for non-aeruginosa Pseudomonas.
Okay, cool. Good to know.